In this lecture, we're going to continue our focus on the cell membrane and examine some additional features of the cell membrane, and we'll conclude this particular video with some examples of these very important membrane proteins. And using these values here just to illustrate the point of how the cell membrane functions to separate the extracellular and intracellular fluid. We'll take a look not at every single item here, but just some of the key ones, particularly when we talk about bioelectrical signaling among neurons or within neurons. So if we look at some of the key players, the sodium ion, the potassium ion, and the calcium ion, we see that sodium is held in much greater concentrations outside the cell as compared to the interior of the cell, and this is all assuming the resting state of a cell. Potassium is opposite, where there's a greater concentration of more potassium ions inside the cell than outside, and calcium has a much greater concentration outside the cell by many orders of magnitude than is found in the cell cytoplasm. And these ions are not the only ones, but are important ions that establishes something called the resting membrane potential, which is a separation of the electrical energy state between the inside of the cell and the outside. Three more examples here. If we take a look at molecular oxygen, carbon dioxide, and pH, the uppercase P represents the partial pressure of molecular oxygen. And we see that the partial pressure of molecular oxygen is greater outside the cell than inside the cell. And so we might think that cells that are particularly aerobic functions by using molecular oxygen. And so that makes sense in having what's called an oxygen gradient from outside to inside. So as the oxygen molecules are brought into the cell, they're being used inside the cell. So the overall pressure inside the cell of just oxygen is going to be lower. And the difference creates that pressure gradient to ensure that oxygen flows into the cell. Likewise with carbon dioxide, since the interior of the cell is the location where carbon dioxide molecules are produced, that pressure favors the movement of carbon dioxide from the interior to the outside of the cell. And so with respect to oxygen, there's what's called a net influx driven by the pressure gradient. In a similar manner, but in the opposite direction, there's a net efflux of carbon dioxide from the interior to the outside that is generated by the pressure. And we see that the interior of the cell is slightly acidic, indicated by the lower pH value, because the interior of the cell is the site for acid production. When we think about bioelectrical energy generation of neurons or other types of cells, membrane permeability is an important understanding here, because membranes are selective in that certain items are allowed in or allowed out of the cell. And membrane permeability refers to the net rate of diffusion or the movement of a substance through each unit area of membrane for a unit. This is the symbol for concentration difference on either side. And there are some factors that certainly affects that diffusion of a molecule through the actual membrane. The first is the thickness of the membrane. So some membranes are thicker than others, and so with an increased thickness, that will decrease the diffusion rate because the molecule or ion has to traverse a longer distance. In terms of moving through the phospholipid component of the membrane, molecules that are more lipid soluble have that greater solubility and can form favorable and reversible chemical bonds with the fatty acid tails of the interior portion of the cell membrane, and so that gives rise to a greater diffusion rate. Membranes that are embedded with more protein channels. Remember, the protein channels, just generally discussed so far, will allow certain items to move through the channel and into the cell or out of the cell, depending upon the ion. So with a greater number of channels, there's going to be an increased rate of transport if there are additional pathways for the membrane or for the ion to move. Diffusion, particularly when we talk about diffusion, we talk about movement through the phospholipid portion of the cell membrane, tends to increase in direct proportion with temperature. And the molecular weight of the diffusing substance does have an influence, is inversely related, where V in this very simple equation, just to illustrate the point, refers the velocity of thermal motion is inversely related to the square root of the molecular weight of that particular item. So essentially, the heavier the item, the lower the quotient, which is the velocity. Things will move slower if they are larger. One important function of neurons that we'll see in this unit is called exocytosis. And so exocytosis refers to the movement of molecules or ions from the interior of the cell to the exterior of the cell through a particular mechanism. So one of the functions of taking an interior item inside the cell and moving to attempting to extrude it to the exterior of the cell is to actually produce membrane components, where molecules to the actual membrane are coming from the vesicle interior. One example is a protein that is stored inside muscle cells, skeletal muscle cells, called GLUT4 or glucose transporter type 4. So this is a protein that sits inside a vesicle, and here are just some drawings of these circles that would contain GLUT4. And given a particular signal, particularly that would be insulin, the cell will move the vesicle to the interior of the membrane and embed GLUT4 into the actual membrane, but not entirely kick it out into the exterior of the cell. Exocytosis also functions to recycle any items, molecules that were brought in the opposite direction, which is endocytosis. And so the cell will take recycled products or molecules and repackage and will tend to add those to the cell membrane or may completely extrude those items to the exterior of the cell. Secretion is another important function where the cell will just release, for example, a neurotransmitter to the exterior of the cell. So some examples of secretion come from a particular type of white blood cell that will release what are called antibodies for immune system function. Neurons will release neurotransmitters. One such example of a neurotransmitter is acetylcholine. And some neurons or some other cells, not just neurons, will secrete mucus. And this is typical of what we see in the respiratory and digestive systems. So here up on the diagram on the left, here we see the circles, which are secretory vesicles, and signals will cause those secretory vesicles to move towards the interior of the membrane, will fuse with the membrane, and in fusing with the membrane, the vesicle actually breaks apart and the interior components will be released into the extracellular region of the cell. This is an electron micrograph of what's called the neuromuscular junction that we'll address in a later video. But here you see the connection between a terminal motor neuron. This is actually the terminal axon portion of a motor neuron, and a muscle cell or muscle fiber. We know this is a muscle fiber because on the left bottom portion of the screen here of this image, we see this very regular appearance of what are called striations. And these are the contractile proteins responsible for force production. The separation, the small tiny gap separation between these two cells is called a primary synaptic cleft. And the muscle cell membrane, this is a skeletal muscle cell. What you see here is a folding of the muscle cell membrane forming these invaginations, and the distance between the terminal axon and to the membrane inside the folds is called the secondary synaptic cleft. And what's illustrated in this micrograph are what are called vesicles. And these are the very small components that contain the neurotransmitters. So you can see a very large population of these vesicles that will eventually fuse with the terminal axon, with the membrane of the terminal axon. And you can see that illustrated here. On some of these locations. And when this happens, they will release neurotransmitter that will cause an excitation of the muscle cell. So this will end our second video here on some of the finer points of the cell membrane.